Okay, so for my protein project, I was assigned the Cras protein, and the mutation was Cras G12C. And so the normal function of this the normal function is as a relay molecule, as this is what determines the growth of a cell, and it relays signals about the amount of growth that can happen at any time. So this means that it is relaying the information to tell a cell whether or not to grow and how much it can. And it's typically off as to not stimulate too much cell growth, but the mutation makes it so that it is always on. And so this protein is found in the membrane cells, as it has to send signals to them on if they need to grow or not, as I said. It is found in all mammal cells because they have to be able to control cell growth. So this would mean it's found in humans, cats, dogs, tigers, any mammal like that. And this is because, again, it's a receptor, it's in the membrane, cytoplasm. It's used to make sure and regulate cell growth. And so the mutation is a single point mutation, and it basically makes it so rather than the active site typically being off with the ADP, it has a mutation where the guanine is substituted with cysteine. I can't really pronounce them. And so this makes it so that rather than having GDP typically, which would make the cell normally off and then at some certain points on, it is only active with GTP. And so this allows cells to grow at rates that it usually cannot, and also grow when they should not otherwise grow. This is the main effect of this mutation, and why it is linked to so many cancer cases, as the rapid cell growth and uncontrolled cell growth is the basis of cancer and how it spreads. And so the functional group of the original chain is glycine, in which it is hydrophilic, neutral, and also the smallest amino acid out of all the 20. And the interaction that glycine has is that it has the ability to have the reaction that will turn GTP into GDP, which allows for the quote-unquote like on-off switch. And this is due to glycine being hydrophilic, so it's able to do the hydrolyzing reaction, in which will turn the ATP, which is the activative, into ADP, which is the off. All while the mutation turns the glycine, which is supposed to be in 12, the 12th slot out of the 188, into cytosine, in which cytosine is hydrophobic, neutral, and then relatively small compared to other amino acids. But the original glycine, again, takes part in the reaction that will convert the active GTP into the inactive GDP in order to limit the amount of cell growth. But when the mutated, it has cytosine, which is hydrophobic, so it cannot hydrolyze the GTP into GDP, which basically means that rather than having an on-off switch, it's always on, which causes the cell to have no limits for growth, and it is constantly given the okay to grow without bounds. This, of course, makes it very dangerous, as it can be deadly for the organisms due to high rates of cancer that can come out of this mutation, which obviously will eventually kill them, typically. And so this mutation makes the protein function unbalanced, as rather than only turning on when needed and then turning off most of the time, it is constantly on and does not turn off when needed. This makes the cell grow out of control when it doesn't need to, which result in the death of the cell, cancer, or the cell losing its intended purpose. So this can affect the entire organism, as if enough of their cells turn cancerous, the organism can die, and if the organism cell get too large, it can get sick or die. And so basically it's just impacting the whole organism. And so for my 3D model, you can see it. Hand's kind of blocking it. But I did the first 30 out of the 188, in which I also have this key. I'll get it in a shot. And so as you can see, as you can see, the overall shape is spherical. I tried to make it as best as I can, but it's very difficult to bend this wire, so I wasn't really able to make it perfectly, but it is cylindrical in shape. And so I used copper. I had the Play-Doh balls for amino acids, and I used copper wire to represent the peptide bonds. And so the subunit of it is a small GT paste, which is represented by the mutation. the mutation is this, the black. This is very important. And then, of course, subunits being the amino acids themselves, in which each ball represents an amino acid. Like this would be the cysteine instead of the thymine. This would be the A. And so, basically, this mutation is a point mutation, which occurs right here at the black. So it'll only affect one area of the protein, although unfortunately this is whether or not it's going to be activated or inactivated form. And so I only chose one model due to it only being a point mutation, so I would make two of these just to show that this one thing changes. So rather I put it in black to bold how it changed, and that's basically everything else is fine, it's colorful, it's wonderful, and then black as if it's cancerous, so it's bad. And so the mutation of Cras causes it to always be activated rather than mostly deactivated, and so this obviously promotes cell growth, and that is why this specific mutation is present in nearly 20% of all cancers and causes cells to grow out of control. So I represented this by having the black ball rather than what actual amino acid it is, which would be glycine, and of course the mutation makes it so it's cysteine or cysteine. And so one thing is that, of course, it doesn't have all 188 amino acids, but that's just because, like, logistically, it wouldn't really work. Because even with this, like, it's still difficult to handle. And so I only did 30 as to, like, show how it is just this one mutation that ruins this whole thing without having to put all of them. And so, of course, I tried to mimic the shape as best as possible, but I'm not able to do it. As, of course, I have 30 instead of 188, so I have to be less exact and not very specific. Thank you.